Hi, we're going to continue talking about plasmids and bacteria as we're kind of getting to our genetic engineering topics. So using plasmids. So we already talked about plasmids. Plasmids are really cool because I can essentially genetically engineer them, and I can make them whatever I want them to be. I can have sites put in them that make insertion easy. I can give them antibiotic-resistant genes like we talked about before. I can give them inducible markers and stuff like that. So there's a couple more things about them. So they have special abilities that make them useful to us, right? So I can use them for gene storage, right? Storage or replication. So a couple things. So the recombinant DNA, right, must be in the form of cDNA, and the C means complementary, right? So cDNA lacks introns. Because remember, bacteria are prokaryotes, and prokaryotes lack the mechanisms to edit mRNA, right? So you as a eukaryote, you produce pre-mRNA, and then it's edited, and the introns are removed, and the exons stay, and then we can make protein products from translation out of the mRNA, right? So to produce these, right, we have to use an enzyme called reverse transcriptase. Remember, transcription makes mRNA, so reverse transcriptase makes back DNA, but it makes it without introns, right? When I store it in a plasmid, now it's called a cDNA library. So we can buy genes that are already inserted into plasmids, right? We just have to look up in a cDNA library, find the gene we want, order it within the plasmid we want, and then we can express it, right? So when I'm storing or replicating recombinant DNA in plasmids, the plasmid is called a cloning vector. Remember, plasmids are vectors. If you see this picture right here, plasmids are vectors, right? Depending what I want to use them for is the kind of plasmids here. So we're going to concentrate on these two, this one and this one. But we have other type of ones too, right? If I'm using it to produce protein products, it's going to be called an expression vector, right? So, for instance, if I put the insulin gene in, right, if I put the cDNA version of insulin inside E. coli, right, and then I go ahead and freeze that E. coli, that's a cloning vector at this point. Once I put that E. coli into my bioreactor and I start to harvest insulin product, now it's essentially an expression vector. Right, we can also use them for modifying the host genome. And we'll talk about this a little bit later, right? As in gene therapy or making GMO plants or different experimental therapies. So, but we're going to concentrate on the top two. So advantages of using plasmids, of storing DNA, right? So as far as linear and plasmids are more stable because they're not linear. They don't have open ends, right? They're a circular chromosome. So they're more stable. That's an advantage, right? Plasmids can be inserted into bacteria efficiently. So a linear piece of DNA has to be incorporated in the main genome, or a plasmid, because it's circular, it has its own ORI site, and it can just start to express itself once it's inserted in the bacteria, right? Plasmids can be engineered to add extra restriction site and be modified, right? So we talked about before commercially, many have what they call MCS, or multiple cloning sites, for ease of gene insertion. We're going to talk about plasmid design in a little bit, but those multiple cloning sites ensure that our sticky ends are ones that we want. Remember, I want to have two different restriction enzymes, theoretically, to improve insertion efficiency, right? So I found this cartoon, right? When we can engineer things called promiscuous plasmids, Promiscuous, generally speaking, if I have a plasmid from E. coli, it can only infect other E. coli's, right? Because it's designed by E. coli's for E. coli's. A promiscuous plasmid is one that I can insert in many different types of organisms, right? So I thought that was interesting. Anyway, one thing that when I'm designing my plasmid, I have to be able to regulate transcription. What that means is, and the important part is this first part right here, the start codon, right, for my gene must be downstream from a promoter. So if I had my plasmid here, right, I would have my promoter, right, and then I would have my MCS site, right, and then I would have my gene, and then I would have my terminator sequence, right? So I have to set it up correctly so I don't get reading frame errors, right? And that's really all that we need to know for that. Okay, transcriptional regulation of plasmids. So in order to be able to change from a cloning vector to an expression vector, we use things called operons. So an operon, right, was originally discovered in E. coli, and what it does is it hides facultative genes, right? It hides facultative genes that only need to be used when the conditions are right. Right, so bacteria are small, they don't have unlimited resources, so for managing resources, helps them survive better. Right, so these are naturally occurring control units that induce or activate or repress or turn off groups of genes. Right, so there's a couple things that we need to know here. So some components that we need, somewhere in the DNA, I have a regulatory gene. So the regulatory gene produces the regulator. Right? So the regulator hides the promoter. Right? So when you see a P here, right? So if you see that orange box there, the P is the promoter. The O is the operator. Right? So the promoter binds RNA polymerase. Remember, RNA polymerase makes mRNA, which we use to make proteins. The operator binds the repressor. So the repressor is made by this regulatory gene, which is not in the operon. Right? And that regulatory gene, right, so here we have this blue-looking Lego thing here, and that's my lac repressor, right, so my lactose repressor. So once I make this, and that's what we call constitutive expression, so it's always being expressed, right? We're always making active repressors, and the repressor will bind to the operator. Now when it binds to the operator, now RNA polymerase cannot get to the lac genes, the lac Z, the lac Y, the lac A genes, right? They make different enzymes, but it can't get to it. All right. So the repressor is then activated by the inducer. Right? So the inducer here is arabinose. Oops, I made a mistake. So the inducer here, we use an arabinose inducer in our lab. But for this one, right, it's allactose, which is a form of lactose. So, and I'll tell you about how it's regulated below, but if the sugar lactose is present, right, it'll bind to the repressor and it'll inactivate it. So lactose right, by changing its conformation. Right, so it binds to it. Remember, that's allosteric inactivation, right? So it binds to it, it changes its conformation, so it doesn't fit the operator anymore, and it falls off. Once it falls off, RNA polymerase is free to transcribe that whole unit, right? So let's talk about how it's regulated. So it's regulated a couple different ways. Now, if I design a plasmid, right? So this is up in the upper right-hand corner. It's a plasmid that's been designed. You'll notice I have a couple things. I have the AMP-R gene at the top. So this is my AMP-R, right? So my ampicillin resistance. I have my ORI, my origin of replication. And then I have my LAC, right? So there's my LAC promoter. I have my RBS, my regulatory binding site, right? So that's where my promoter would be, right? My operator. My regulatory binding site is going to be my operator, right? And then I have my MCS. So I have my multiple cloning site right in there. You'll notice I have another regulatory binding site. And then I have my LAC gene down in there. And then I have a terminator sequence. So they're designed. So the way that I can regulate it then, right, is by managing growth media. Right, so let me move this over here a little bit. Let's try to make this a little smaller. Okay. All right, so this is the way it's regulated. So if I have the LAC operon here, so there's a couple things that we didn't talk about. So I have my P, it's my promoter, right? So this is my promoter. That's the binding site for my RNA polymerase. I have my operator, and this is my regulatory gene over here. Usually I have an I, right? My LAC I, so my lactose inducer gene, right? So that's my regulatory gene, right? And it sits on top of the operator and it's blocking transcription. All right. I also have, next to the promoter, I have a cap site. So the cap site change colors on that because it's getting all. In fact, I'm going to move it all the way over here. So my cap the computer's going crazy. So it facilitates the binding of RNA polymerase. That's what the cap does. All right. So the cap facilitates the binding of RNA polymerase. All right. So this is activated. By CMP or CAMP, which is cyclic adenosine monophosphate. And here's the reason why. So if I look at this picture over on the other side, right, here's my lac operon. So I've got my cat protein, right? So if glucose is not available, but lactose is available, remember lactose is going to cause the repressor to fall off the operator, right? And then I'll have cyclic AMP. So ATP, so here's the skinny of it. ATP, this is made from glucose. Right? So I use ATP, remember, with energy. So if I'm using ATP, I'm going to get ADP plus an inorganic phosphate, right? Plus energy. Right? I can use this and I can get MP plus PI energy. So if I have glucose is present, I'll just go and make more ATP from it, right? In fact, if glucose is present, I'll never get to the AMP. I'll just take the ADP and make it back into ATP, right? Adenosine diphosphate, adenosine monophosphate, right? But if glucose is not available, right, then I'll get to this AMP. Now, I can't go any farther than AMP, but AMP, otherwise known as cyclic AMP, right, stimulates the production of energy, right? So what happens is, if I don't have glucose, because bacteria just can't go find glucose, right? If I don't have glucose but lactose is available, the cyclic, so we'll put a C here, the cyclic AMP will bind to that cap area. Yeah, it keeps locking up my pen. So it'll bind to that cap area, right? And it'll facilitate the binding of RNA polymerase, and now I'll go ahead and make enzymes and I can start making ATP again, right, from the lactose, because it'll make lactose into glucose, and then I can do that glucose to make more ATP. If glucose is available, I don't get cyclic AMP. Because I don't get cyclic AMP, this cap won't allow RNA polymerase to bind, right? If I have low glucose, but lactose is unavailable, the repressor, right, so this is my repressor here, right, my cyclic AMP is there, but my repressor still won't allow it to be, allow the lactose to be digested, right? Because I'm not going to waste, the bacteria is not going to waste the energy, right, to convert lactose into glucose, right? If there's no glucose or there's no glucose. If I have high glucose and high lactose, right, this cap binding protein, right, still sits on here and blocks RNA polymerase. So the only time, right, that I can express my protein is if I have low glucose but I have lactose available. So if I make cloning vectors and I want these cloning vectors to replicate, right, without making my protein product, then I just give it glucose and I don't give it any lactose, right? If I want to start upping production of my protein, then I put it in a bioreactor with low glucose but an abundance of lactose. Then it turns on that mechanism and starts pumping out my protein. So I can manage these operons by managing the operon unit and the food source, right? And you'll see that's what happens here in this bottom picture, right? When I have cyclic AMP, right down here at the bottom, right? It binds to the cap protein so it comes off. And then once it comes off, it exposes the promoter, and RNA polymerase can go ahead and attach to it, right? And then go ahead and make proteins. Right. So engineering. So I just want to show you, here's an example of the P-glow. So we use the green fluorescent, right? The P-glow is incorporated with the P-bad operon, right? Or what I get is the R-bad operon. So the P-glow fluorescent marker is incorporated into the R-bad operon, otherwise known as the P-bad, right? So if you look at the P-glow, right, I have a RC, right? So that's the arabinose operon. I got the P-bad, the arabinose promoter. So I've got my my RC is my regulatory protein, right? So this right here is making that gene's making the repressor. I got my arabinose promoter, so this repressor binds to the operator. Right? And then I've got my green fluorescent protein, which is under the control of PBAT. So I can't make the GFP, right, unless I can turn on the promoter site. And I have BLA, which is my ampicillin resistance gene in my ORI. So everything I need there. Right? So if I expand, I can expand this a little here, and we can look at that P-bad operon. Right? So here's an expanded view of it, because I want to show you what we have here. Right? So we have our, like we said, we have our RC. Oops. All right. So like we said here, we have our R. Man, I don't know why that keeps going with it. Let me try one more time. We have our RC there, right? So that's making our repressor, right? I have my operator, so the repressor that it makes is going to bind to my operator right here, right? We have the RC promoter, right? So that's where my RNA polymerase is going to bind to. I've got another operator here I can control. I've got my cap site, remember, that I can turn it on and off with cyclic AMP, right? And then I start to have different things. So I have a restriction site here, right? So I can cut it open and put something there. I've got another promoter. I've got my arabinose promoter right here. And then here is my MCS site. I've got all those different restriction enzymes. I've got my eCore1, I've got my BAM here, I've got my Hindi there, right? So I've got all these common MCSs. And then when we come over here, I have my BLA here, my BLA. That's my antibiotic resistance gene. But you'll notice here, I have all these other genes here, right? I have these other restriction sites here, right? So these ones here, the MCS is in my operon unit, but I can also insert other genes that I want turned on all the time, right? And then I've got my ORI, right? I've got an origin of replication over here, and there's another type of ORI over there. Okay, so all our components are there. Right, so this BAM is turned on in the presence of arabinose. Like I said, it includes the BLA gene for antibiotic resistance. And really the thing about the operon is it allows scientists to check for proper insertion, and we can control expression. All right, well, hopefully that was informative, and I'll talk to you soon.